Hi everyone, and welcome to the Micro e-learning program from the Global Parkinson's Genetics Program and the Aligning Science Across Parkinson's. My name is Mariam Isayan, and I'm a neurologist at Somnus Neurology Clinic in Yerevan, Armenia. During this model, we will briefly discuss what GWAS is and how it is used to identify genetic factors associated with Parkinson's disease risk, age at onset, and progression. While the exact cause of Parkinson's disease is yet to be uncovered, it results from a complex interplay between genetic and environmental factors. Genome-wide association studies, or GWAS, are used to identify genetic regions associated with a phenotype of interest in ancestrally similar, unrelated individuals. GWAS works by testing allele frequency differences to pinpoint statistically significant genetic variants linked to a particular trait or disease. To ensure sufficient study power, GWAS requires large sample sizes. As you can see in this plot, the gradual increase in the number of cases leads to a higher number of identified loci. The larger the sample size, the smaller the magnitude of effects and frequencies you are able to accurately identify. Prior to conducting a GWAS, genotyping data undergo quality control, which includes excluding low-quality variants, identifying sex inconsistencies, and estimating genetic relationships among individuals. In GP2, the GenoTools package is used. Related individuals at the level of first cousins or closer are excluded. Data dimensionality reduction techniques such as principal component analysis or PCA and uniform manifold approximation and projection or UMAP are used for population stratification, followed by imputation, a process that enriches genotyping array data by inferring missing variants based on linkage disequilibrium with genotyped variants. These QC steps are crucial for increasing the statistical power of GWAS and are discussed in detail in modules 1 and 2 of the GP2 Beginner Bioinformatics course on the GP2 Learning Platform. GWAS involves performing regression analysis for each variant, adjusting for covariates such as principal components for population substructure, sex, age, and other relevant factors. The choice of association test depends on the outcome. Linear regression is used for quantitative traits such as age at onset, while logistic regression is applied to binary outcomes like disease status in cases and controls. Summary statistics from GWAS include outputs like p-values and association metrics for each variant. In logistic regression models, the odds ratio represents changes in the odds of binary outcomes, while in linear regression, the beta coefficient shows the estimated effect size of genetic variant on a continuous trait. These summary statistics are commonly visualized in plots such as Manhattan plots, and they also serve as input for further meta-analysis. Several recent studies have reported Parkinson's disease risk loci across different ancestries. A GWAS meta-analysis by Nals et al. Focusing on individuals of European ancestry identified 90 independent Parkinson's disease risk loci, including 38 novel signals. In a study by Fo et al., 11 significant Parkinson's disease risk loci were identified by comparing Asian and European ancestries. Among these, 2 novel loci were found exclusively in Asian individuals, while the remaining 9 had previously been identified in Europeans. A study by Loci et al. Focusing on the Latin American population identified the SNCA locus as reaching genome-wide significance, with list SNP RS356225. Additionally, 82% of the variants identified by Nals et al. Were concordant in their effect size direction. In the study by Rizek et al., a novel GPA1 intronic risk variant was reported as a common risk factor for Parkinson's disease and age at onset in individuals of African and African admixed ancestry, while being rare in other populations. A functional study by Alvarez Jerez et al. Showed this non-coding GPA1 variant was associated with decreased protein expression through affected splicing, reducing glucocerebrosidase levels and activity. In contrast to the aforementioned studies that analyzed single populations, Kim et al. Conducted a large-scale multi-ancestry GWAS meta-analysis, including individuals of European, East Asian, Latin American, and African ancestries. The meta-analysis identified 78 independent genome-wide loci, where 12 were potentially novel loci, and fine mapping resulted in 6 putative causal variants with known 6 Parkinson's disease loci. Age at onset in Parkinson's disease contributes to the clinical heterogeneity of the condition. Early onset forms of Parkinson's disease significantly impact the overall burden of the disease and are often linked to monogenic causes, while sporadic Parkinson's disease cases can manifest at various ages. Common genetic variants associated with Parkinson's risk contribute to earlier onset of disease. According to the study by Nals et al., cumulative burden presented as a genetic risk score of 28 variants reported at the time of the study was strongly associated with earlier age at onset in typical Parkinson's cases. The largest GWAS on age at onset in Parkinson's disease conducted by Blauendrath et al. Identified two significant loci in SNCA and TMEM175, each associated with a reduction in age at onset of approximately 0.6 years. Disease progression in Parkinson's disease is variable and is often assessed using scales such as MDS-UPDRS, Hoehn-Yahr, MMSE, and MoCA. In a study by Carrasco et al., a large GWAS on Parkinson's disease motor progression was conducted using data from MDS-UPDRS Part 3. The study identified the GGA5 locus significantly associated with the progression of symptoms affecting gait and posture. In a study by Liu et al. That focused on identifying genetic variants influencing the progression from Parkinson's disease to Parkinson's disease dementia using MMSE data, the RIMS2 locus was found to significantly affect cognitive decline in Parkinson's disease alongside GPA and APOE loci. Another study by Tan et al. Focused on clinical and motor progression variants using the MDS-UPDRS and Hoehn-Yahr scales. They identified several significant independent genetic signals associated with cognitive decline, including APOE. Additionally, a novel signal in ATP8B2 was associated with motor progression, although it did not reach statistical significance. A GWAS by Iwaki et al. Revealed that significant variant in SLC44A1 was associated with faster progression to severe stages of the disease. Additionally, known risk variants in the GPA1 and APOE loci were confirmed to influence both motor and cognitive decline. In conclusion, GWAS aims to identify risk loci associated with binary or continuous outcomes such as an increased risk of developing Parkinson's disease, influencing the age at onset and the progression rate of both motor and non-motor symptoms. Recent studies underscore the global impact of GWAS, revealing genetic risk loci across different populations. Both multi-ancestry and single-ancestry GWAS meta-analyses have uncovered novel genetic variants specific to various ethnic groups, thereby enhancing our understanding of disease and genetic predispositions towards precision therapeutics on a global scale. Thank you for listening, and I hope you enjoyed the video.